Hi, this is Dawit and this is my Osmosis egg lab video. In this video, I will show my egg, who I named Egna, and her 5-day transformation. The purpose of this lab is to see the diffusion of water in different tonicity solution. We will see the egg's shape change, difference in tonicity, concentration gradient, and the volume and turgor pressure. I started by taking a container that would fit Egna and filling it with vinegar until Egna was fully covered. I did this at 10 a.m. on the weekend, which led to a small error in my experiment. The next day, when I was supposed to replace the vinegar after 24 hours, I couldn't, because I would have been at school at 10 a.m. So I replaced Egna's vinegar after only 21 hours. This could have affected how well the calcium carbonate egg shell dissolved, because it was sitting in the acidic vinegar for a shorter amount of time. This also could have interfered how well my egg was going to absorb the water and syrup, because the shell might have still been present, making it harder for the ingredients to pass through. Luckily, no major changes occurred in my experiment. Throughout the hours, Egna began to bubble in the vinegar and the shell started to dissolve, leaving a foamy white residue in the solution. After 48 hours, I had my naked egg. It was jelly-like and had a semi-transparent appearance. I rinsed Egna and let her dry on a paper towel as I cleaned my container for the syrup. I used maple syrup and covered Egna completely in it, and during this step, I calculated my time correctly so that my egg could sit in the syrup for a full 48 hours. Over time, Egna became shriveled, and wrinkly. At this point in the experiment, Egna was sitting in a hypertonic solution. This meant that there was a higher concentration of solvent in my egg cell. The syrup is so sweet and sugary because it has a high concentration of solute. In a hypertonic solution, water moves out of the cell due to a higher solvent concentration in the cell. This left Egna with a flaccid shape because there was no solvent to keep the cells in shape. There had been a decrease in volume which lowered the turgor pressure in the cell. If I kept Egna in the syrup for a longer amount of time, she might have gone through cronation, which is animal cell death due to the lack of cytoplasm to function. This part of the experiment is very similar to a grape and a raisin. A raisin loses water, which makes its shape shriveled, but it tastes sweeter because there's a higher concentration of solute, which is sugar. Unlike a grape, which has a full shape because it has a higher concentration of water, but a less sweet taste. After 48 hours, I rinsed Egna and filled my container up with water that I dyed blue. I placed Egna into the water solution and observed it after a few hours. Egna had grew and no longer had her flaccid shape. Egna was now in a hypotonic solution. This meant that Egna was sitting in a solution that contained more solvent than the cell. In this solution, the net H2O moves into the cell, going from a high concentration to a low concentration. This made Egna's shape larger because there was an increase in volume, which also increased the turgor pressure of the cell, because there was more water to keep the shell's shape. Again, if I left Egna in this solution for much longer, she might have gone through cytolysis, which is animal cell death due to such a high turgor pressure that the cell explodes. This is very similar to why grocery stores spray their leafy vegetables with water. Leafy greens tend to wilt and don't look that appealing to buy. So when they get sprayed with water, the plants take in a high concentration of water, which helps them look fresher due to the plant cells being more full of water. Through this experiment, I got to see the different tonicity solutions, volume and turgor pressure, and potential cell death. Osmosis is an important passive transport as it moves water molecules from a low solute concentration to a high solute concentration across a membrane that is not permeable to the solute. And I got to see that through this experiment. I hope you enjoyed watching my video of me learning about osmosis and applying my knowledge to this experiment. Thank you.